Hello immunologists! Those of you who have already watched the viral immunity lecture will have noticed two things. Firstly, that it was filmed before a live studio audience, and secondly, that there's no mention whatsoever of SARS CoV2 or COVID-19. And that's because the lecture was filmed last year. Now, obviously, under the circumstances, we can't not talk about COVID-19, so welcome to this bonus lecture. A few things I want to mention before we begin. Firstly, I'm going to assume that you're familiar with many of the basic principles of viral immunity that I went through in the recorded lecture. So if you haven't watched that yet, please do. Secondly, this is an incredibly fast-moving area, and everything I'm going to tell you is up to date as of the 8th of May, which is when I'm recording this, to the best of my knowledge at least. But new information is coming to light all the time. Finally, I'm going to show you data from a lot of preprints. These are scientific papers that haven't yet been through peer review. Now, the ones I've chosen to show you are, in my view, good science. But if you go looking at preprints yourself, do bear in mind that they haven't yet had the scientific community's seal of approval, so you might need to be a little bit more critical than you usually would. So today we're going to begin by talking about the biology of the virus. And because this is an immunology lecture, I'm not going to delve too deeply into the virology. But just like in our viral immunity lecture, we are going to consider the viral receptor, the cellular receptor, and the nature of the viral genome and how that might impact things like the mutation rate, which we know is important for how long-lasting immunity can be. Then we're going to talk about the immune response to acute infection. And just like before, we'll consider how we sense the virus. Interferon is very important here, and then how we respond. And there I'm going to consider as well aspects of immunopathology, including why old people seem to get sicker. Then we'll consider what happens after a person recovers. How long might we expect them to be immune for? And finally, I'll give you an update on where we are with vaccines and immunotherapies. So let's start with the virus, which is called SARS CoV2. This stands for Severe Acute Respiratory Syndrome Coronavirus 2. The first SARS coronavirus, of course, being the one that caused the 2002 outbreak. I was at school at the time that happened, and it's also what set me on the course that a couple of years later took me to university to study viral immunology. So the virus is SARS CoV2. The disease is COVID-19, which is coronavirus infectious disease first identified in 2019. You'll hear lots of people calling the virus COVID-19, which isn't strictly correct, but it's so widely done that everyone understands what it means, and I've even heard virologists doing it. So this is an enveloped single-strand positive sense RNA virus. The envelope is the reason that it's so sensitive to detergents. Washing your hands with soap and water really does help. And the fact that it's an RNA virus might get us started thinking about whether, like flu, we might expect to see antigenic drift, and how that might affect the longevity of the immune response. We'll talk about that a bit later. Now, the reason that this family of viruses is called coronaviruses is because of this rather lovely crown shape that we can see under the electron microscope. The spikes of the crown are made of a structural protein called the spike or S protein, which I'm showing you here. It's both the viral receptor and it seems to be the main immunogenic component of the virus for raising an antibody response, which we'll talk a bit more about later. The cellular receptor is angiotensin converting enzyme 2, or ACE2, and it's found on lung type 2 alveolar cells, enterocytes in the small intestine, endothelial cells and arterial smooth muscle cells. This is the lung of someone who died of SARS-CoV-1, which also uses ACE2 as its cellular receptor. You can see ACE2 in green and virally infected cells in red. And as we discussed in the viral immunity lecture, the cells that express the viral receptor determine the viral tropism. That's to say which cells the virus can infect. And this is the reason that we see SARS-CoV-2 infection mostly in the respiratory tract, but also in the intestine. And we can potentially use knowledge about the viral receptor to design treatments that block viral entry. But since this is an immunology course, the only ones that I'm really going to talk about today are the ones that use antibodies. So the spike protein is the most studied, but the virus has a total of 29 proteins. There are 3 other non-structural proteins, 16 non-structural proteins, which are required for the virus life cycle but not actually included in the virion, and then these accessory proteins, which are not needed for the virus to grow in cell culture, but are required in the host. And as soon as you see protein that has those properties, you should straight away be thinking, what's in the host that's not in a cell culture dish? It's the immune system. So it's likely that these are immune evasion proteins. I said in the viral immunity lecture that we can tell a lot about the aspects of the immune response that particularly bother a virus by looking at the immune evasion proteins that it encodes. So let's try and use that to work out which particular bits of the immune response are important here. The coronaviruses that we know most about are the mouse model virus, the model virus mouse hepatitis virus, and the human viruses SARS CoV2 and MERS. And they all seem to be particularly bothered by the type 1 interferon response. SARS-1 has a whole load of proteins, non-structural proteins 10, 13, 14 and 16, all of which act to guanosine cap and 5' methylate its RNAs, which means that they resemble host RNAs and won't be recognised by RIG1. MERS has a protein NS4, which binds to dsRNA, shielding it from MDA5. And mouse hepatitis virus has a protein NSP14, which is a double-stranded DNA exoribonuclease, which again seems to interfere with this part of the pathway. MHV mutant viruses that lack NSP3 can only infect interferon receptor knockout mice. So that's another viral immune evasion protein that works in the interferon pathway. And MERS, NS4B, and mouse hepatitis virus NSP2 both cleave activators in the RNase L pathway, interfering on the receptor cell side of the interferon response. And we also know that some interferon-induced genes called iFTIMs act to prevent fusion of the viral envelope with the cell membrane in SARS and MERS, preventing their entry. So it may well be the case that something similar happens here. So if we use the model that we used in the general viral immunity lecture, how does the immune system sense the virus and how does it respond to the virus? We can see from the immune evasion genes that the interferon pathway is likely to be really important for sensing. And this paper that looked at the genes being transcribed in the lungs of COVID-19 patients compared to other forms of pneumonia shows that the interferon pathway is really activated in these patients, supporting the idea that the interferon pathway is important for detection. We also know that pre-treating cells with type 1 interferon in vitro prevents SARS-CoV-2 entry, and that in mouse and monkey coronavirus models, including models of SARS and MERS, having a delayed interferon response is associated with more severe disease. Now, this is a small study, but it's quite intriguing. We've got 3 COVID-19 patients in France early in the outbreak. Patient 1 is critically ill, and patients 2 and 3 are brought into the hospital because they're contacts of patient 1. Patient 3 gets severely ill, but patient 2 remains asymptomatic throughout. And notice that it's patient 2 who makes a good interferon response. So a robust interferon response may well be protective. To know exactly how we respond is a bit trickier because generally people turn up at hospital when they get sick, which is often weeks after they were infected. So we don't usually have the opportunity to look at the early immune response. Also, a lot of people never get sick enough to go to hospital. They may not even be symptomatic. So we also don't have the opportunity to look at mild infections, except perhaps in that handful of studies that happened early in the outbreak when there was still capacity to bring in contacts of people who are infected. But what we can do is we can compare immune readouts for healthy, moderately and severely sick individuals, and that does tell us something. And I'm actually going to start with this one from very early in the outbreak, which is just one patient. But she wasn't that sick. She recovered by herself without the need for any intervention other than a bit of intravenous fluids. Also, the doctors looking after her took readings at a number of time points, so we can see the development of the immune response over time. So this lady attended hospital 4 days after her symptoms started, and we think probably about 2 weeks after she caught the virus. And you can see the state of her lungs on the first day that she was in hospital, which is about 5 days after her symptoms started. But by day 10, she's pretty much recovered. And you can see her TFH and antibody producing cells come up in this period before she recovers. you can also see activated CD4 and CD8 positive T cells coming up. And here are the monocytes and the NK cells, the activated NK cells. The monocytes seem depressed throughout, even after she's recovered. Although since this is just one person, we really can't be sure if that's an effect of the disease or something specific to the patient. But from all of this, the authors of the paper conclude, probably fairly, that since we see the adaptive response kicking in before she gets better, and that her doctors didn't give her any treatment, it's because of the immune response that she got better. So in moderate, and probably also in mild cases of the disease, the adaptive immune response is helpful, which is what we would usually expect. But as we start to compare moderate and severe cases, we start to see that the immune response isn't necessarily all good. Here we can see the immune cells and cytokines and how they're altered in severe disease. So in severe disease, neutrophils go up, and the lymphocytes and the monocytes go down. And I'll show you in a minute that this is probably because they're being recruited to the lungs. We also see very high IL-6, IL-8 and IL-10. A number of studies have shown that IL-6 levels at admission are the best predictor of who will go on to get severe disease. So you can see that in general, severe disease is associated with more inflammation and probably more recruitment of monocytes and T cells to the lung. And in this paper, they looked in a little bit more detail at the T cells and the monocytes. I'm showing the CD4 positive T cells here, but they saw the same among the CD8 positive cells. And all these markers of activation are increased in people with severe disease compared to those with moderate disease. These cytokines are being produced by the T cells at higher levels, but also the T cells are expressing more markers of exhaustion. We also see the monocytes making these cytokines, although interestingly in this study, the patients with more severe disease had more monocytes, not fewer. And over a whole bunch of these papers that have characterised the immune response in the blood, which I won't show you all of, people do disagree a bit about whether the numbers of monocytes go up, down, or stay the same in severe disease, but they do all agree that the monocytes are activated. Finally, here's a paper that used single-cell RNA sequencing to look at the cells in the blood of COVID-19 patients in an unbiased way. So because they're not using flow cytometry, they're not limited by which markers they choose to look at. And they're only looking here at 7 patients and 7 controls, which might limit what they can detect. But still, they managed to see significant changes in activated granulocytes, IgA and IgG producing B cells, and a decrease in NK cells, which might suggest that the NK cells are being recruited to the lungs. And when they looked in more detail, they found that the monocytes displayed a signature of genes consistent with activation by interferon gamma, and that in some people, but not all, the NK cells did too. Well, that's the blood, but what about the lungs? Well, looking at bronchoalveolar lavage fluid using single-cell RNA sequencing again, we can see recruitment of NK cells and T cells in patients compared to controls. But actually, you can see that patients with mild disease have more CD8 positive T cells than the severe patients, which might suggest that the CD8 positive T cell response in the lung is protective. The difference in NK cells that we see between moderate and severe cases wasn't significantly different, so I don't think that we can make any conclusions based just off that. But while we're talking about NK cells, I will mention that in a mouse model of SARS infection, if you deplete the NK cells, the mice get sicker. So that suggests that NK cells, like CD8 positive T cells, are likely to be helpful. But if we start looking at the macrophages, we see this big group that are present in healthy patients, and these represent lung-resident macrophages, and it's probably broadly anti-inflammatory. But it's down in the moderate patients and even further down in the severe patients, whereas these 3 groups, which represent inflammatory macrophages, are really very expanded in the severe patients. So it looks like the recruitment of monocytes, which will convert into inflammatory macrophages, may be a bad thing. And this is backed up by the observation that if you deplete macrophages in a mouse model of SARS, the mice get less sick. So clearly a deal of the pathology is immune pathology, and we call this particular pattern of immune pathology a cytokine storm. And it may have a bearing on a feature of the disease that I'm sure you've all heard talked about, which is the fact that the case fatality ratio increases with age. And this was also a feature of SARS and MERS. So why does this happen? Well, one idea that was floated very early in the outbreak, when it was still mostly confined to China, was that there's some kind of antibody-dependent enhancement. This is a phenomenon by which an antibody which isn't perfect, for example, an antibody that was raised to a related virus, or, for example, if you maybe have a low titer of antibody for some reason, can promote the uptake of the virus by cells like macrophages, which then move it around the body, spreading the infection. And there's good evidence that this occurs in dengue, and it's thought to be the reason that it's the second dengue infection that gives you the hemorrhagic fever. There's also some evidence in MERS that antibody can cause spike protein to alter its conformation, making it easier to enter its ordinary target cells. So one idea was that older people might have met something like SARS CoV before, and the antibodies against it were causing them to become sicker, as with dengue. I think that's probably not true, for two reasons. Firstly, because if it's due to a cross-reaction to another coronavirus, it's difficult to know what that virus is. It can't be SARS or MERS, or we wouldn't be having problems in the parts of the world where those viruses didn't spread widely. And it's unlikely to be one of the 4 human coronaviruses that cause colds, because if it was, the at-risk population would be young adults and up, rather than, as it is, the over 65s. Another suggestion was that older people have a generally impaired ability to respond to new antigens, which is broadly true, and that's one challenge of developing vaccines for them. But for this disease, older people actually develop a better antigen-specific response, possibly because they get sicker, and sicker people also develop a better antigen-specific response. But still, I think we can probably rule that one out. That's the adaptive response. But what if they have a problem in their innate response? Well, in mouse and monkey models, and older animals are slower to make a type 1 interferon response. And slow interferon responses are associated with a higher likelihood of cytokine storm later in the disease. So I think this possibility is still in the running. Another is that older people tend to have low-grade systemic inflammation, and that possibly this higher starting point for inflammation could make it easier to tip them over into the edge of a cytokine storm. One way we could perhaps get at this is that lots of these same changes happen in people who have HCMV, which we talked about in the viral immunity lecture. So if that's the case, we might expect people who have had CMV, well, who still have CMV because it's a herpes virus and it goes latent, we might expect people who have CMV to be more likely to get severe disease. I can't find any official data on that yet, but I have looked to see if countries that have higher rates of CMV seroprevalence have had worse outbreaks of COVID-19, and that doesn't seem to be the case. But there are so many confounders, not least how well each individual government handles the outbreak, that I don't think we can lean too heavily on that kind of analysis. And if someone looks at whether individuals who have had CMV are more susceptible within a population, I think that would be very interesting. Finally, because we're immunologists, of course we're looking for immune answers to this, but we should consider the possibility that it's nothing to do with the immune response. For example, we're beginning to get reports that people with underlying lung conditions have higher levels of the viral receptor ACE2, which might mean that it's easier for the virus to infect their cells. And in the livers and guts of hepatitis C patients, ACE2 expression increases with age. So that could be the reason that older people are more susceptible. And this data might also suggest that it could be one of the reasons that men are more susceptible, because they also seem to express more of the ACE2 cellular receptor. So that's what we know about the immune system in acute infection. But what about as the infection resolves? Do we become immune? But before I talk about that, I want to talk a little bit about how we measure whether someone is or isn't immune. That's going to be important for things like the idea of immunity passports, where the government will allow you to go back to work if you're immune. Although I think it's worth mentioning that the immunologist who gave evidence at the select committee hearing on this idea last week thought that even our best tests probably aren't good enough to do that yet. But we do all agree that being able to tell if a person is immune will be important for vaccine trials, because one of the ways that we work out if our vaccine has worked, other than seeing if it actually protects people against the disease, is to measure things that we think correlate with protection, like antibody response. Well, all over the world, for all sorts of infections, we work out if people have had a disease by doing an ELISA for IgG, usually in the serum. And for more than a month now, we've actually had a number of workable ELISAs for antibodies against the spike protein, most of which work something like this. We coat the tissue culture plate with spike protein and allow it to stick for a couple of, well, usually overnight actually, wash off anything that's not stuck. Then we're going to add serum from our potential immune person, and if they have made an antibody response to the spike protein and they've got IgG against it, that will stick to the spike protein. We wash off what hasn't stuck, and then we add an anti-human IgG antibody, which is conjugated to horseradish peroxidase. Wash off what hasn't stuck, and finally we add the substrate for the horseradish peroxidase, and if everything is lined up like I've shown you here, then the colourless substrate will change colour. And we can effectively see how much someone has got anti-spike protein IgG in their serum by seeing how blue the well goes. This particular test was developed by Florian Kramer's group, and I mention them specifically because they were really quick at turning their lab over to this as far back as January, which was visionary really, because they couldn't have known then that this would become a pandemic. But I also mention them because they've been absolutely exemplary in sharing their reagents and techniques so that labs all over the world could get this assay set up quickly. But these are lab tests, and one of the things that the government has really wanted to have and has sunk a lot of money into is point of care testing. So you might have heard about these pregnancy test type devices, and the proper word for this is a lateral flow test. And they're supposed to work something like this. You put the sample, which will be serum or blood, onto the sample pad, and if you have anti-SARS CoV2 antibodies, they will move with everything else onto the conjugate pad, where they will attach to spike protein, which has been covalently attached to something that will show up as a colour if it is grouped together. In this case, it's going to be colloidal gold. And as your complexes of anti-SARS antibodies and SARS-CoV antigen coated in colloidal gold continue to move up, if there's the IgG there, then that will get caught by anti-human IgG on the IgG line, or anti-human IgM, if you've got IgM, on the IgM line. And finally you have a control line which basically detects an extra antibody that's been put onto the test just to check that the flow is actually going in the direction that we expect. So that's how it's supposed to work, but there have been a number of problems with them. So as a general rule, lateral flow tests are about 100 to 500 times less sensitive than ELISAs, and that's mainly because you're looking at a couple of minutes incubation rather than a couple of hours. And that can be good enough, it works for pregnancy tests after all, but certainly these tests are going to need a lot more work if they're ever going to be useful. The other problem is that if you get a very high titer of antibody in the sample, you might start to get immune complexes which interfere with the result. So these tests might not work either at high or at low concentrations of the antibody, and certainly they're not going to replace ELISAs anytime soon. At the moment, they're only able to identify about 65 to 85% of positive cases, which certainly isn't good enough. So both of these tests will tell you if there's antibody, or at least they're trying to, but not if the antibody is neutralizing. So if you want to know that, you have to see if the patient's serum can prevent the virus from infecting cells. But working with live virus is really quite dangerous, so people are using retroviruses that have been pseudotyped with the viral receptor. That means that they've been engineered to express SARS-CoV2 spike protein instead of whatever viral receptor they would usually use. And these pseudotyped viruses would normally be able to infect a cell line expressing the cellular receptor ACE2. What you do then is you add patient serum, and if it contains neutralizing antibodies to the spike protein, that will prevent the virus from entering your cell lines. And so if you see cell lines that don't get infected when you've added patient serum, that tells you that the serum contains neutralizing antibody. So if we start to look at patients with these kinds of tests, what do we see? This heat map summarises the percentage of patients who have antibody by day since they started to have symptoms in a cohort of about 500 patients, with red being more people and blue being fewer. And you can see that on average people are starting to make some kind of response at about day 10 after their symptoms start. And as we would expect, we get an IgM response before we see the IgG response. This paper also showed that people who had more severe disease made a better antibody response, which is similar to what we saw in MERS. If we instead look specifically at neutralizing antibody using that viral infection assay, you see the same sort of thing, with neutralizing antibody beginning to emerge at about day 10. Now this study was quite interesting because it had 175 people in it, but 10 of them didn't make any neutralizing antibody at all, even though they did recover. Well, maybe they were making non-neutralizing antibodies, or maybe antibody isn't actually the thing that protects us. Maybe it's just a good indicator that correlates with something that actually is protective, perhaps T cells. Which brings me neatly onto how we'll measure the T cell response, which we do using a technique called an ELISpot, which works something like this. We take whole immune cells and we're going to pre-stimulate them with antigen. And because we're looking for a T cell response, we're usually going to be looking at how they respond to antigen that's being presented by an antigen presenting cell. And then we transfer them onto pre-coated plates, let them stick down. Then we add a biotinylated anti-cytokine antibody for T cells, usually interferon gamma. So that's going to stick to any interferon gamma that the activated T cells are making. And then we add developing reagents that the biotinylated antibody will cause to change colour. And we'll see spots forming wherever there's a T cell that is specific for whatever antigen you stimulated the cells with. So here's an ELISpot looking at T cell responses to SARS CoV2. You can see that we have spots in the recovered COVID-19 patients, but not in the controls. But also notice that we're getting T cell responses to different parts of the virus than the antibody responses. We're getting responses to the nucleocapsid and membrane proteins rather than the spike. And that makes sense because T cells will be seeing the viral protein being presented on MHC, whereas the B cells are seeing native protein. So that's why the T cells are able to recognise these other proteins that are hidden within the virion. So we're seeing people raising these T and B cell responses, but one big question is how long will these responses be good for? And that's important because there's the idea that we could let people who've already had the virus back out, but that will only work as a strategy if they're not going to get infected again. So this is an RNA virus, and you'll remember from our viral immunity lecture that RNA-dependent RNA polymerases that these viruses use to replicate are error-prone. And you'll also remember that the main barrier to long-lived immunity in influenza is that it's continuously changing in a process called antigenic drift. So will SARS CoV2 do something similar? Well, the good news is that it does seem to have a lower mutation rate than the flu. And if you look on a gene, because unlike the flu, its polymerase does seem to have some proofreading ability. And if you look on a gene by gene basis, it's particularly stable in the spike protein, probably because if that mutates too much, the virus won't be able to infect host cells. I should also state explicitly, since we're comparing with the flu, that because the genome is non-segmented, we don't need to worry about antigenic shift. And skipping ahead for a moment to the idea of a vaccine, a preprint that came out last week used this kind of analysis to suggest that if we can come up with a vaccine, the virus is antigenically stable enough that we probably won't need to keep changing it, and we won't need to include different substrains. And that's in contrast with flu. So from all of this, I would estimate that the virus will be antigenically stable for at least 5 or even 10 years if our protective immune responses last that long. But will they? Well, when we think about viral immunity, we have a tendency to think about viruses like measles, mumps and rubella that before vaccination you would get once and then you would be immune for life. In the 1846 measles epidemic on the Faroe Islands, everyone got measles except for those people who had been alive in the last epidemic, and they were still protected 65 years later. But we do have viruses against which the protective immune response is very short-lived. For example, if you get the winter vomiting virus, you're protected for 12 or maybe 16 weeks. So which of these extremes is SARS CoV2 likely to be closer to? Of course, it's really too soon to tell, but we can look at other human coronaviruses, and in MERS, as well as SARS, we do lose antibody titers over the course of years. But of course, as I've already said, antibody might not be the be-all and end-all, and this study found in recovered SARS patients found that they could detect antigen-specific T cells 5 years later. So maybe we do have some long-lived T cell immunity. On the other hand, studies looking at coronaviruses that cause colds in humans suggest that that's probably not the case. In a handful of studies where people were experimentally infected with these relatively harmless coronaviruses and were then re-challenged 12 or 18 months later, they wouldn't get symptomatic infections. Although note that in one study people did get infected, just asymptomatically, which would obviously be a problem for SARS CoV2 since we know that asymptomatically infected people can and do infect other people who can then get very sick. So we have a year or so's protection at least, but if we take a look at the epidemiology of these viruses, the rate at which young people get infected is so high that if we were then immune for life, we would expect adults to never catch these viruses, which we know that they do. So all of this suggests that if we don't come up with a solution to this situation, we're going to have recurring epidemics of this disease going forward. And of course, as immunologists, the most obvious solution is a vaccine. Now, ideally, we'd want one that you could vaccinate people with once, maybe give them a booster, and then they'd be protected for life. But even if we can't develop a vaccine that does that, if we can make one that gives people a couple of years protection, that could work. Or even if we could just develop something that would reduce the severity of the disease. Both of those things are roughly true, after all, of a vaccination strategy that we use against seasonal influenza. So when will we have a vaccine? The first thing to say is that we actually do have a number of veterinary coronavirus vaccines, some of which can elicit long-term immunity even though the viruses themselves don't. So that's hopeful. But there are also some lessons from veterinary coronavirus vaccines that might give us some pause for thought. Most of the vaccines that are successful are against gut viruses, but there is one, just one, against a respiratory virus, which is infectious bronchitis virus of chickens. And there are both attenuated and inactivated versions of that vaccine. And 2 of the SARS CoV2 vaccine candidates are inactivated virus. I suspect that we don't have an attenuated vaccine candidate yet because that will take at least a year of passage in eggs or in culture, and we're obviously looking for a much quicker solution here. But the problem with both the attenuated and the inactivated vaccines in chickens is that they don't give a good mucosal IgA response, so they don't actually protect against infection, although they do reduce the severity of the disease. And it might be that we have to settle for a similar situation, at least to begin with the human SARS CoV2 vaccine. The second veterinary coronavirus that we can perhaps learn something from is the feline enteric coronavirus, where despite many efforts, a vaccine hasn't been possible, largely because all the experimental candidates have caused antibody-dependent enhancement of viral disease. So that's something that we'll need to be alert to when we do our safety trials of the SARS CoV2 vaccine in humans. So what's the human vaccine situation? Well, the first thing to say is that there almost isn't a form of vaccine delivery that isn't being tried here, except, as I said already, an attenuated vaccine. Anyone who can make anything is having a go using their favourite method. Now, the graph I'm showing you here was published on the 8th of April, showing the 115 vaccine candidates we had on that date, broken down by approach. And the last date that I can find information for is the 20th of April, at which point we still had 115 vaccine candidates. So there may be a few more since then, probably not too many more, but even if there aren't, 115 vaccine candidates is a lot. And I'm giving you the details of some of the front runners in this table. But this information is changing rapidly. And the website that I've taken the table from is updated every couple of weeks. So if you want to stay up to date, I would suggest having a look there. One thing that I notice is that a lot of the candidates are adenoviral vector vaccines, including 2 of the front runners. And basically the approach here is to engineer an adenovirus to express COVID-19 protein. And then when you give it to people, you hope that you'll get the benefits of a live viral vaccine, which will usually give you a more type 1 immune response. And that should be more protective. I hope you remember that the type 1 immune response is the main one that protects against viral diseases. So you should get those benefits without the disadvantages of having to spend a year attenuating the real virus. But I should be clear that no adenoviral vector vaccines have yet been approved, although a couple of the emergency vaccines that were produced for the Ebola outbreak were basically on the verge of approval. Another vaccine that I think is worth actually mentioning is this one, which is a recombinant protein vaccine that uses an old-fashioned adjuvant. It's really cheap, about $10 a dose, but it's actually a vaccine to SARS-1 that was developed between 2011 and 2016. It's very safe and protective in mice, and the team were ready to take it into humans. Even they had the doses ready to give people manufactured to the correct standards for human use. But then their funding dried up, so they weren't able to take it into trials. But as it happens, antibodies from mice immunised with the vaccine do cross-react with SARS CoV2. So the team are going to see if this works in humans. And the reason I mention this one is because I think there are 2 important lessons here. Firstly, perhaps if we had continued to fund vaccine development against other severe coronaviruses, even when the threat seemed to have receded, we would have been in a much better position now. But also, this is a pandemic. And if we want to get the disease under control on a global scale, we're going to need cheap, stable vaccines like this that can be supplied to everyone in every country, not just people in wealthy countries. So the vaccine trials are all going to have to focus on 2 things, safety and efficacy. For safety, we'll want to see that people don't have any very serious side effects straight away. Serious reaction. And for both of the approaches that I've talked about just now, past experience tells us that we can be fairly confident that that won't happen. But a lot of the things that could go wrong, we won't know about for a little while. One is that if the pathology is immune mediated and we boost the immune response, we might see people who receive the vaccine getting worse disease. Another is if we raise an antibody response that promotes antibody-dependent enhancement, again, we might see people who receive the vaccine getting worse disease. And that did happen with some of the SARS-1 vaccine candidates. So it's something we've been really aware of this time. Another thing that could happen is if we accidentally raise a response to a host protein, then we'll get autoimmune disease. And again, we won't see that for a while. This is actually what happened with the emergency flu vaccine that was produced in the 2009 pandemic. One of the rare side effects was narcolepsy, which is actually an autoimmune disease. Of course, the other side of the coin is, will the vaccine protect people against the virus? And there are a number of ways to approach this question. An easy one to see is to see if you get good antibody responses, which we've already mentioned do seem to correlate with protection. But it doesn't guarantee protection against the actual disease. For that, we're just going to have to wait to see if the volunteers catch it. There have been some ideas about speeding this part up by experimentally infecting people. I don't think that's likely to happen, but people are doing these kinds of experiments in animals. One issue I can see with the adenoviral vector approaches is that previous vaccines that have used this approach have usually needed to give a boost with another form of vaccine in order to get good protective immunity. And we're going to have to see if that will be the case here. As far as I know, none of the trials have a boost step planned at the moment. So finally, while some immunologists are developing vaccines, others are developing immunotherapies. So how might some of these work? Well, these approaches all rely, one way or another, on antibodies. And the lowest tech of them is simply to give patients plasma from someone who has recovered. And that will contain a whole mix of antibodies. This is an approach that we already use for other viral diseases. So, for example, chickenpox is very serious if you're pregnant. So if a pregnant woman who hasn't had chickenpox or the vaccination herself is exposed to someone with chickenpox, we'll give her pooled IgG from a whole bunch of other people, most of whom will have had the chickenpox. And that protects her and her baby. We haven't yet got any proper clinical trials on this with proper control groups, but the small uncontrolled studies that have recently been published tell us at least that the approach doesn't seem to be harmful, which it might have been if there was some kind of antibody-dependent enhancement, and possibly it's beneficial, although without a properly controlled trial, it's hard to tell. These trials are being rolled out. There are 54 of them registered now, and so there's a big push for recovered plasma donors in order to have the materials to do these trials. So higher tech approaches use monoclonal antibodies, and we do now have neutralizing monoclonals to SARS CoV2, which have been identified by sorting B cells whose BCRs recognize the spike protein. How do you find these B cells? Well, you make a fluorescent spike protein, allow it to bind to B cells, and then you sort those B cells that are bound to the fluorescent protein. And you PCR out the B cell receptor, transfect it into other cells, allow them to produce antibody, and then you see if that antibody that the cells are making prevents virus entering cells. And taking that approach, the authors of this paper found 3 neutralizing monoclonal antibodies. But this approach is actually being taken by teams all over the place, and particularly by biotech firms whose main business is in making monoclonals. And there are trials of those due to start in the next couple of months. Finally, there are a number of trials that aim to alter the immune response. We've talked a lot about the idea that a poor or late interferon type 1 response might be associated with more severe disease. And as of today, there are 33 trials that are trying giving patients interferon to see if that helps with the disease. That's an approach with a very long pedigree in the treatment of hepatitis B and hepatitis C, and it also works well in mouse models of SARS. We mentioned that T cells in COVID-19 patients seem to be more exhausted, expressing higher levels of PD-1. So what if we block PD-1? Will that reactivate the T cells? 6 trials are trying that. So these approaches are trying to activate the immune system. But if immunopathology is the problem, might we actually want to inhibit aspects of the immune response? I mentioned before that IL-6 is one of the main components of the cytokine storm. So one obvious question is, can we therefore make disease less severe by blocking IL-6? And 49 trials are trying that now. Oops. A few reports came out suggesting that IL-6 does bring people's fevers down very quickly, but none of the studies that have come out so far have had proper control groups, so it's difficult to tell, and we're going to need to await the results of properly controlled trials. People are also trying some of the immunosuppressive drugs that we talked about in our transplantation lecture, with 28 trials looking at corticosteroids, 5 trials looking at drugs that in a transplant situation we would classify as the anti-proliferative component of the immunosuppression. And we even have 4 trials looking at some of the humble non-steroidal anti-inflammatory drugs such as ibuprofen. If you'd like to keep up with how these trials are going, there are a couple of global clinical trials dashboards. I like the one that I've given you down at the bottom right because it classifies the drugs by their mechanism of action, which makes it much easier to navigate. So we've come to the end of the lecture, and at this point I would usually give you my take-home messages. That's really tough under the current circumstances because the situation is changing so rapidly at the moment and there's still so much that we don't know. So instead I'm going to invite you to come and continue the conversation over on Blackboard, and thank you for listening.